Before we dive into the physiology of muscle hypertrophy, it's important that we begin with a quick review of muscle cell structure and function. And as we progress through this course, you're going to want a basic understanding of how muscle cells function so that you can comprehend how its structure adapts from the different mechanisms of hypertrophy. To really grasp the notion of hypertrophy, you must be able to recognize what works, why it works, and how it works. So here's a classic textbook diagram of a cell, and it's important to remember that cells come in all different shapes and sizes based on their unique function. And the essential characteristic of skeletal muscle is that it voluntarily shortens or contracts. Skeletal muscle is the only type of muscle that we can continuously control through our nervous system. And here's an image of a relaxed bicep at full length compared to a contracted bicep that is fully shortened. And on the right, you will see a diagram of the sarcomere contraction. Muscle cells have a long cylindrical shape, and because of this, they are often referred to as muscle fibers. So remember when you're learning about human physiology that muscle cells and muscle fibers are used synonymously and basically mean the same thing. Now, connective tissue binds muscle fibers in a hierarchical pattern. First, the entire muscle is enveloped in a sheath of fascia called the epimysm. And the second layer, the muscle is broken down into what are called muscle fascicles, and these fascicles are all enveloped in a sheath of connective tissue called the perimysm. And then within these muscle fascicles, we have individual muscle fibers, and all of these fibers are connected by connective tissue known as the endomysm, which is the smallest layer of connective tissue. So here's a 3D image of the connective tissue. And all three layers of connective tissue can be referred to as fascia. And you can clearly see the thin outer layer over the muscle, the white layer in between the muscle fascicles, and the little bit of white in between the individual muscle fibers. The health and elasticity of the fascia can play a major role in hypertrophy, and we're going to touch on that in a little bit. An individual muscle fiber at the cellular level has several parts. We're going to focus on two main parts and how they relate to hypertrophy: the myofibrils and the sarcoplasm. The sarcoplasm is a gel-like substance that envelops the space in between the myofibrils. So if you're looking at this image, you'll clearly see a myofibril on the top image. And then you'll see the sarcolamina, which is the casing all around the muscle fiber. The space between, the off-white looking space, would be the sarcoplasm. It would be the fluid within the muscle cell. Here is another 3D image of a muscle fiber. And this image, you can clearly see the myofibrils and the sarcolamina. And if you could imagine filling the space in between the myofibrils with a gel-like substance, that would be the sarcoplasmic fluid. And this gel-like substance stores nutrients and a significant amount of glycogen and myoglobin, which is an oxygen-binding protein. If you look at the lower image, you'll see something labeled the sarcoplasmic rectilium. Be careful not to confuse it with the sarcoplasm or sarcoplasmic fluid. The sarcoplasmic rectilium is the tubular portion that almost envelops the myofibrils, and it's responsible for controlling the flow of calcium, which is responsible for muscle contractions. Here is an oversimplified diagram of the sarcoplasm. It's as simple as just looking at the space that's around the myofibrils inside the sarcolamina. The myofibrils are the rod-like portions of the muscle fiber, and they're composed of long proteins that help hold them together. A muscle contracts by sliding the thick myosin protein and the thin actin protein filaments along each other. And here's an oversimplified diagram of the myofibrils. They're simply the red dot within the cell. So let's talk a little bit more about how a muscle cell functions. Muscle fibers can be broken down into two main types: slow twitch type 1 muscle fibers and fast twitch type 2 muscle fibers. Type 2 muscle fibers can subsequently be broken down into two more categories: type 2A, which is often referred to as fast twitch oxidative glycolytic, and type 2B, or sometimes called type 2X, which is often referred to as fast twitch glycolytic. Slow twitch or type 1 fibers are referred to as slow twitch oxidative. And these type 1 slow twitch fibers are used in lower intensity exercises, such as very lightweight resistance work aimed towards muscular endurance, or long duration aerobic activities like a 5 or 10K run. Type 1 fibers are identified by having a slower contraction speed and being structurally smaller, meaning they have a smaller neuron and a smaller fiber diameter. They're also considered highly resistant to fatigue because they have a high mitochondrial and capillary density and a high myoglobin content. This high myoglobin content, along with an extensive blood supply, gives the muscle fiber a red color. Type 2 muscle fibers, also called fast twitch muscle fibers, produce a high peak force in a relatively short period of time, meaning they have a much quicker contraction speed. Now, type 2B fibers are very prone to fatigue because they produce the most force when stimulated. And these are the last fibers recruited when a muscle gets activated. And they're also the first to stop being recruited when the force from the muscle is no longer needed. These type 2B are often called type 2X and have the largest muscle fiber diameter, which makes them ideal for hypertrophy training. Type 2A fibers are the intermediate fibers. Their peak force and their resistance to fatigue fall between the two classifications of type 1 and type 2B. So these fibers are recruited after type 1, but before type 2B. And they stop being activated after type 2B, but before type 1. What's neat is that training can influence these fibers to behave more like type 1 or more like type 2B, depending on the type of training. You're born with a certain ratio between these fibers, and your ratio can affect how successful you are at either developing as an endurance athlete or a power athlete. Most people have a 50% type 1 and 50% type 2 ratio, but many elite athletes, meaning world-class runners or Olympic sprinters, can have up to 80% of one or the other type. So, for example, a sprinter with 80% fast twitch fibers will have a much better chance at being an elite competitor than somebody with only 30% fast twitch fibers. Your muscle fiber composition is built into your genetics, but only to a point. Like I said earlier, training can influence type 2A fibers to behave more like type 1 or type 2B. So, with periodizing training, practice, and time, you can pretty much be a competitive recreational athlete in any sport. But to really be an elite level, generally you want to move towards what your genetic disposition is in terms of your fiber type. Since hypertrophy is the name of the game, we're going to look at some individual muscles and their common muscle fiber ratios. because some muscles have a predominance towards one type of muscle fiber. It is unlikely that it will change enough to show a completely different dominant fiber type. So, for example, your soleus is on average 80 to 96% slow twitch fibers, and it's highly unlikely that excessive and extensive training will change it to be predominantly fast twitch fibers. So, training for maximum muscular development will most likely require more focus on the dominant muscle fiber type that already exists. So, for the example of your soleus, you may need to favor more high rep endurance work to optimize hypertrophy. So, we're going to take a look at the lower body first. And if you look at the hamstrings are on average 55% type 1. So they're leaning towards more slow twitch, and the gluteus maximus is 56% on average type 1, meaning that they're also leaning more towards slow twitch. The soleus ranges between 80 to 96% type 1, meaning it's very slow twitch dominant, whereas the gastrocnemius ranges between 50 to 76% type 1. And if you're leaning on the side of 50%, you may be able to train your soleus more slow twitch and your gastroc more fast twitch. So in the gym, this might look like seated calf raises for higher repetition ranges and standing calf raises for lower repetition ranges. Now, when we look at the quadricep, we can see some muscles with fast twitch tendencies. So for example, the rectus femoris has a slight tendency towards type 2 muscle fibers, with studies reporting it having a range of 30 to 50% type 1. So it can be anywhere between 50% fast twitch to 70% fast twitch. And the vastus lateralis seems to average around 42% type 1, meaning it's pretty close to 60% fast twitch. So it would appear that a lot of the lower body postural muscles have a tendency to be slow twitch, with some of the non-postural muscles having more of a fast twitch composition. Now, when we look at the upper body, things are a little bit different because there's not as many postural muscles. So the pectoralis major is mixed fast twitch, roughly 38% type 1, meaning it's almost 40 to 45% fast twitch. And the latissimus dorsi is somewhere between 30 to 50% type 1, meaning it's anywhere between 50 to 70% fast twitch. The triceps are about 40% type 1, meaning they're about 60% fast twitch, whereas the deltoids are one of the few upper body muscle groups that displays a tendency towards a higher proportion of type 1 muscle fibers. It's on average 58% type 1, so it's only about 40% fast twitch. We're going to briefly touch on motor units now and dig a little bit deeper into it when we cover mechanisms of hypertrophy. Motor unit recruitment refers to the activation of additional motor units to accomplish an increase in contractile strength in a muscle. And the higher the recruitment, the stronger the muscle contraction is going to be. Motor units are generally recruited in order of smallest to largest or slow twitch to fast twitch, as contractile strength is needed. Simply put, we recruit our smallest and weakest slow twitch fibers first, and then continuously add larger type 2 fibers until we can generate enough force to overcome an exercise. So if you look at the image on the left, it shows each motor neuron attachment to individual muscle fibers, which is a part of one motor unit. Meaning, if you look at where it says red type 1, and you see all the attachments spawning off into the red muscle fibers, and it all comes down into that one motor unit. And in this image, there's two red type 1 motor units and 2 blue type 2 motor units. Now, the far right image shows a real-life microscopic model of motor neurons attached to individual muscle fibers. Okay, so if you're a Seinfeld fan, you'll love this meme. Remember earlier when we mentioned how fascia could play a role in hypertrophy? Fascia, like muscles, can become tight over time and restrict range of motion as well as room for muscles to grow. Remember, there are 3 layers of muscular fascia: the endomysm, the individual fibers, the perimysm, the fiber bundles, and the epimysm, which is the sheath over the entire muscle. The theory is that your muscles can become trapped and bound in a tough interconnected network of fascia, which could restrain muscle cells from expansion and inhibit hypertrophy. So let's just do a summary of everything we covered in this module. The sarcoplasm is the space between the myofibrils, and it's filled with a gel-like substance that stores nutrients called the sarcoplasmic fluid. The myofibrils are responsible for muscle contraction. Muscle fibers, also called muscle cells, vary in types and can be affected by training. Those types are type 1, slow twitch, and then type 2A and type 2B. Motor units are responsible for muscle activation and force generation. And finally, fascia can restrain muscle cells from expansion and possibly inhibit hypertrophy.